Okay, it's my pleasure to introduce Dr. Aleksey Zimin, who's been working in the field of bioinformatics since 2002, starting at Celera Genomics, also known, of course, for being involved in the first human genome sequencing project. And now he's work. And now he's working at Johns Hopkins University at the Department of Biomedical Engineering. He develops algorithms and software for de novo genome assembly and annotation for the latest generation of sequencing data. For example, he has developed the Mazurka genome assembly package and used it in assembly of more than 2,600 genomes. He's also the leading author on widely used tools such as Mummer4, Polka, and Jasper and Samba scaffolder. And today he's going to talk to us about his novel automated genome annotation package called EviAnn. Aleksey, the word is to you, and thank you again for accepting our invitation.
Good day, and thank you for inviting me. Thank you for your kind introduction, Stephen. So today I'll be telling you about my latest project is development of EviAnn, which is short for evidence-based annotation. And it's ultra-fast, user-friendly, evidence-based genome annotation software that's being developed by our team at Johns Hopkins University. What is gene annotation? Well, gene annotation, especially protein coding gene annotation, solves two main problems. You have to locate transcripts on the genome, that is, locate sets of coordinates of exons and introns. And then for each transcript, you have to decide whether it's protein coding transcript or a long non-coding RNA, or it's just transcriptional noise, it needs to be discarded, that you get from the data. And then within each transcript that you decided that it's protein coding, you have to correctly locate open reading frame or coding sequence, which is abbreviated as CDS within the transcript. And generally, annotation is presented in GFF format, GTF or GFF format, which basically gives you sets of coordinates on the genome where genes start and end. Within the genes, there are mRNA structures, which these are transcripts. And within each transcript consists of a set of exons. The GFF file will give you start and end of the exon. And within if it's a protein coding transcript, within the transcript, you also get sets of coding sequences where if this is positive direction, this is where the start codon starts. If it's a complete CDS, that's annotated, and this is where the stop codon ends. So, and getting these coordinates is the problem that annotation software solves. And it helps you make sense of the genome. It locates functional elements, and then it enables a host of subsequent studies. Annotation, there are many ways to approach this problem. So for bacterial genome, you don't really need evidence. They are nearly all sequences coding, and even if you get overlapping genes on opposite strands. So evidence is not really strictly necessary for annotating bacterial genomes. And ab initio gene finders do very well for those genomes. However, for eukaryotic genomes, you have to use evidence because gene space is sparse, so by far not all sequences coding, and there you have to be able to distinguish between functional and non-functional parts of the genome. So the evidence that you can use is ESTs. This is legacy evidence or express sequence tags. They're legacy evidence and it's not being widely used. The most widely used transcriptional evidence is Illumina RNA-seq data. There's also PacBio IsoSeq and now Kinnex protocol that work very well recovering nearly complete and nearly complete transcripts. There is also Oxford Nanopore, direct RNA and cDNA sequencing, and finally protein homology. And protein homology is extremely useful because wherever you are generating a set of transcriptional data such as Illumina RNA-seq or PacBio or Oxford Nanopore, unless you're willing to do lots of experiments sequencing multiple tissues with at multiple conditions, you may not see all transcripts expressed in your libraries, in your sequencing runs. And so protein homology, protein identity is preserved at very large evolutionary distances. And so protein homology may help you recover those transcripts or coding sequences that are not present in your transcriptome sequencing data. I would like to mention the importance of automated annotation. So it's very important to use annotation technique that's been validated. And automated annotation is a validated way to annotate genomes. What this means is that we, developers of annotation pipelines, we test them on a wide variety of genomes, on a wide variety of data sets. And we tell you in our paid publications what to expect from our annotation, from our products, from our software. Once authors in a manuscript decide to use a combination of tools, that's actually akin to designing a novel annotation pipeline, and result may be of varying quality. So every time one uses a set of annotation tools and then somehow combines the results, question arises from my point of view, from a point of view of developer. So this this is in principle a new annotation pipeline, and I would ask, has this new approach, new combination of tools been tested in real life on multiple genomes? And as a result, if there is no testing data, how can I trust this annotation that's been produced by a combination of existing tools? And that's why when we develop annotation software, we show you multiple runs with multiple data, multiple inputs on multiple different genomes, so that you can see the quality of the results that you can expect from our pipeline. Typically, most published annotation pipelines use ab initio gene finding techniques. That is, they use evidence, be that protein alignments or RNA-seq data, and they use evidence to train one or more ab initio gene finders. Then they combine the gene predictions and again use evidence to filter them to eliminate spurious predictions. And then finally, this results in annotation. There are problems with ab initio gene finders. In general, ab initio gene finding is a great approach. It does allow you to recover missing information, that the information of missing in RNA-seq data or incomplete protein alignments, but they can be problematic on eukaryotic genomes. They do give you many false positives. You have to work really hard to filter the data. They're not transparent. That is, imagine you have your transcript that you know is functional, you know it's there, and you put it in as evidence, and it's not guaranteed that ab initio gene finder will output this transcript. It may or may not exist in the output. So it's like a black box. You train it and you get some results, and it's really difficult to figure out why certain transcript has been annotated or not, even though you have confidence and it's been present in evidence. Also, ab initio gene finders typically only find coding sequences, that is CDSs. And if you look at the anatomy of a typical protein coding genes, you may have 3 exons, exons 1, 2, and 3. We have a UTR, 5' UTR, and then here's your coding sequence start codon ATG, and then your coding sequence ends in a stop codon, and there follows 3' UTR, and there may be exons that are all in UTR regions. And ab initio gene finders will typically yield only the shaded area between ATG and TAA. And any exons or parts of exons that are in the UTR regions typically are lost. Well, I ask a question. Can we do without ab initio gene finding altogether? The answer is possibly. However, we have to deal with problems that transcripts derived from RNA-seq data are noisy and incomplete. And another question is that's the answer to which is not straightforward. Okay, even if we get the transcripts right, how do we annotate coding sequences or CDSs within transcripts? How do we find the appropriate open reading frame within transcripts? The idea behind EviAnn is to solve these problems by combining evidence from protein homology and transcript data. I'm going to show you, before telling you how exactly I do this in EviAnn, I'm going to tell you how well EviAnn works. So I'm going to use test data sets for 6 organisms. Most of them are well-studied model organisms, such as Arabidopsis thaliana, fruit fly Drosophila melanogaster, zebrafish Denierario, poplar tree, Populus trichocarpa, chicken, G. gallus, and Mus musculus, which is the mouse. And these test data sets would have 5 to 6 RNA-seq experiments and proteins from 5 to 10 close relatives. And I took these data sets from recently published paper on Breaker3, which fully automated genome annotation tool. And Breaker3 is actually the best, in my opinion, it's the best genome annotation tool. So that's based on ab initio gene finding. And that's our main competitor to EviAnn. And since EviAnn is evidence-based, to avoid possible criticism that I used evidence to make EviAnn look good, I'm going to use exactly the same evidence that was used in Breaker3 paper to compare EviAnn and Breaker3. And in the following slides, I'm going to compare results on these organisms from EviAnn, Breaker3, another popular, however older pipeline Maker2 using SNAP ab initio gene finder following Maker2 protocol paper, Finder, and then finally string tie. So most of these programs, EviAnn, Breaker3, and Maker2, they use string tie assembled transcripts as input. And in my results, I add string tie to show the value that these pipelines provide over its inputs, over the transcripts input by string tie. And in evaluations, I'm going to use GFF compare tools, and I'm going to compare these annotations to RefSeq annotations of protein coding genes and long non-coding RNAs. And I'm going to give you results on sensitivity and precision, with sensitivity defined as a number of features that match reference annotation over the total number of features in reference annotation. So basically, it means what percentage of the reference transcripts we were able to correctly identify. Transcripts, genes, or CDSs. And precision is the number of correct features versus the total annotated features. So if you're being very aggressive and you annotate a lot of transcripts, then your precision will be low and sensitivity will be high and vice versa. There's always a trade-off between sensitivity and precision. And in following slides, I'm going to evaluate quality of transcripts, gene loci, and proteins or coding sequences. And for transcript to match, I'm going to ask for a complete exact match of intron chain, allowing some mismatch on transcription start and stop sites because these are not very well defined. For CDSs, since for those objects, both start, stop, and intron chains are very well defined, I'm going to ask for complete and exact match, including start and stop positions. And then for gene loci, a gene loci is annotated correctly if a tool gets at least one transcript or CDS annotated correctly for that locus, then I'm going to count it as annotated correctly. This is the first evaluation. This is evaluation of gene loci. So again, I give a passing gray mark for gene locus if a pipeline gets a transcript or CDS that matches a reference at that locus. So you only have to get one. And of course, if a pipeline annotates a transcript of CDS or CDS where a reference has nothing, that's a false positive. So here you can see that for Arabidopsis thaliana and Drosophila melanogaster, EviAnn is absolutely the best. For poplar tree, Danio, chicken, and mouse, again, EviAnn provides the best result. The reason I single out Arabidopsis and Drosophila is because in reality, reference annotations are not really perfect. And of these organisms, of these 6 organisms, the best and most complete annotations are actually for Drosophila melanogaster and Arabidopsis thaliana because they are the easiest. They're easier to study, of course, than more complex mammalian or avian genomes. And in my experience, these annotations seem to be the most complete and correct. And here you can see that string-tie does actually pretty well. It can't do CDSs, but it does pretty well in gene loci. And sometimes it even beats Maker2. But of course, Breaker3 and EviAnn do provide a lot of value over the string-tie transcripts that they take as input. Over here, you also see that Maker2 results are missing from Daniel and mouse genomes. And the reason for that is that I had to abort Maker2 run after a month because it was just taking too long. And I'm not willing, I was just not willing to wait for longer than a month for a genome annotation. So Maker2 results will be missing for these particular organisms. And one reason for that is that these particular organisms have lots of proteins in the input data sets, and Maker2 takes a very long time when you have a lot of protein evidence. Next annotation is CDSs and proteins. CDSs or proteins, and CDSs are bread and butter of ab initio gene finders. They are aimed at finding CDSs, and they're pretty good at it. So if you can look at, if you can see over here, Breaker3 does very similar to EviAnn on Arabidopsis and Drosophila and poplar tree. EviAnn outperforms it in terms of on zebrafish, chicken, and mouse. And again, just to remind you, ideal result is here in this corner. So the closer we are to this corner, the better the result is. So here on Arabidopsis, we can say that EviAnn is slightly better than Breaker3. And here on poplar tree, you can see that Breaker3 may be slightly better than EviAnn. That's one data point where Breaker3 beats EviAnn on the same data. And overall, you notice that for these data sets, they're not very big. And the sensitivity of annotation decreases as we go towards more complex organisms, such as chicken and mouse. And final evaluation is evaluation of transcripts. So this was CDSs, this is transcripts. Never mind the CDS, this is wrong on this slide. I should have removed it. So, and transcripts are tougher because you have to be able to get all alternative isoforms. And it happens many times that 2 different isoforms may give you the same CDS, and they may be different by just one single exon in non-coding part. So for transcripts, you see that overall sensitivity and precision are lower. And this is also largely due to transcripts representing long non-coding RNAs in the reference. They are particularly tough to annotate because you don't have accompanying protein evidence. And also you can see that ab initio gene finders based pipelines, they don't do very well on this evaluation because they don't, precisely because they don't annotate UTRs. And overall sensitivity and precision are much lower on the transcript level evaluation just because it's just the hardest to get. So for this transcript level evaluation, you have to be able to get all isoforms at all gene loci right. which is a much harder problem than getting one isoform or getting a coding sequence, just due to degeneracy between transcripts and coding sequences. Well, you may ask, for the mouse genome, the sensitivity and precision is super low. I mean, how can we, this is a very disappointing result. Well, an answer to this criticism is that for mouse genome, this particular data set is just way too small to annotate mouse genome correctly. This data set only has 6 RNA-seq data sets, and this is by far not sufficient for a good annotation of mammalian genome. So to compare, I ran EviAnn with increasing the number of RNA-seq data sets from 6 original data sets. This is this point right here, and right here, to 24. So I did 6, 8, 12, 18, 24. And again, I just got RNA-seq data from NCBI SRA. And here I show you quality of EviAnn annotations of genes, CDSs, and transcripts. And you can see that uniformly the quality of annotations increases as we increase data, as we get more RNA-seq data. Although the increases, we see diminishing return. So over here, you see 6, 12, 18, and 24 just gives you slightly better precision over the same sensitivity for genes. For CDSs, you can see clearly that 24 is the best. You get 6, 12, 18, 24. And in F1 sense, actually, you see uniform improvements. And over here in transcripts, you see a mixed picture, but again, 24, 18, or 12 are much better, give you much better sensitivity and precision than using just 6 RNA-seq data sets. Yes, thank you for the question. UTR annotation seems highly heterogeneous. And is there any post-processing analysis that can harmonize enhanced UTR annotations using RNA-seq data? The only way we can annotate our UTRs is just accept transcripts. They accept what transcript sequencing gives us, and then find the CDS in the transcript. If a transcript has captured UTR region correctly, then we accept we accept it. Unfortunately, there is no better way for us currently to get UTR annotations rather than just taking them from transcript annotations. And what we do actually in EviAnn we compare data between different RNA-seq runs. And if different RNA-seq runs give us different coordinates of the UTR regions, we take an average of that coordinate. So average of the transcription start site and transcription end site. One note that I wanted to mention is Drosophila melanogaster all proteins versus RefSeq proteins. So RefSeq annotations by far are actually not complete. Well, not by far, but they're just not complete. So, for example, if we look at EviAnn and Breaker3 annotations, EviAnn annotates 17,000 unique proteins for Drosophila melanogaster, and Breaker3 annotates about 1,000 fewer. If you look at the proteins in common with unique proteins in RefSeq annotation, yes, EviAnn gets more proteins in common with RefSeq. But then when you add all Drosophila melanogaster proteins that are also in GenBank, you see that some proteins that these pipelines annotate, but that are not in RefSeq, they're actually present in GenBank. And EviAnn gets 196 more proteins that are present in EviAnn annotation, but not present in RefSeq, but are present in GenBank protein databases, and Breaker3 gets 164. And I manually examined some of these proteins, and I did find proteins which are published. For example, this particular protein has a 100% match to a GenBank accession, and it's a well-studied protein and there is a publication about it, and it's absent from RefSeq, and it's present in GenBank, and EviAnn gets it correctly. This particular one, Breaker 3 doesn't get, so that's why I paid attention to this one. A lot of these proteins, of course, overlap. Both programs get these proteins, but this particular one, I wanted to see if since Breaker 3 didn't get it, is it really spurious or was it really valid? Finally, timings of EviAnn. This is a point of our achievement. This is walk log time in hours, excluding time spent on alignment of RNA-seq data, because that's highly variable. That time would depend on how much RNA-seq data you get. Of course, aligning 100 RNA-seq data sets will take you much longer than aligning 10 RNA-seq data sets. But once RNA-seq data, and you have to do alignment of RNA-seq data for all of these programs. So it's a common time. But once RNA-seq data is aligned, EviAnn annotates Drosophila and Arabidopsis thaliana in less than 20 minutes. It takes 1 hour on zebrafish. It takes less than half an hour for poplar tree and chicken, and it takes just about less than an hour for the mouse genome, which is much faster than all other pipelines. And these timings are measured on one single 24-core Intel Xeon Gold computer. So we don't need a cluster, and we don't need any excessive memory requirements. Of course, this performance is largely due to efficient algorithms designed by other people used inside EviAnn. And I would like to specifically note MiniProt, designed by Heng Li. EviAnn uses MiniProt to align proteins to create spliced alignments of proteins to the genome, and that's key in this quick performance of EviAnn. This software designed by Heng Li, and which is MiniProt. Very quickly, I think I'm running out of time, so I'm going to very quickly go over key points of EviAnn algorithm. So EviAnn starts with transcripts built from RNA-seq data, which it by default computes using HiSat2 and string tie2, and actually string tie3 in the latest version. And it also uses alignments of proteins from several related species to the genome, and it computes them with MiniProt. Now, here is the key idea. You take transcripts and you take proteins, and you look for intron chain matches between transcripts and aligned proteins. Most proteins from related species will not match, but some of them will. And if you get an intron chain match from a related species protein to a transcript, you're good. In most cases, this would give you the correct frame. You can extend the match a little bit along the transcript if you are missing start or stop codon, but this would be a golden example of protein coding transcript where a transcript has been confirmed by a intron chain match from a protein from a closely related species. However, you get very few of such alignments, less than half actually of transcripts and CDSs match like this. So these are easy examples, and we use them to train Markov chain model to recognize splice sites, because we are very confident, and from our experience, we know that these transcripts confirmed by proteins, the quality of their intron junctions is around 99.8%. 99.8% of them are good. So we use these intron junctions to train a second-order Markov chain model to then screen splice sites in transcripts that don't have matching CDS, or CDS alignments or protein alignments that do not have matching transcript. And then we reject the CDS alignments or transcript alignments or assembled transcripts that have low Markov chain scores for one or more splice sites. There are also questions when there are also algorithms to deal with inconsistencies. Let's say both transcripts in CDS do pass, but there is no real intron chain match. Some splice sites do mismatch between CDS, between aligned proteins and transcripts, and we do have algorithms to deal with that inside EviAnn to create a combined combined gene model. Some transcripts end up without any CDS matches. There's a very small portion of them. And for those, we have to decide, okay, whether it's when no CDS matches that region, we have to say, look, it's either long non-coding RNA or we just don't get a protein hit. For those transcripts, we look for an ORF, and we use transdecoder 2 for now in our pipeline. And we annotate the ORF if it's found, if it's not found, and if we have a set of transcripts at a locus where we see no ORF in any of these, we annotate that locus as long non-coding RNA locus. We also annotate transcripts. We also annotate some pseudo transcripts that are just CDSs based on protein alignments alone, if they pass our criteria. And finally, and this is the part where we get transcripts that are or rather CDSs only that are not expressed in our input transcriptome data. And finally, we combine results. We have code to detect pseudogenes. We detect process pseudogenes where you see a single exon gene that aligns completely to the interior of another multi-exon gene. That's a process pseudogene, and that typically is non-functional, and we label it as such, and we erase CDS records for that gene. And we also have an option to perform functional annotation automatically by aligning the transcripts to UniProt database, which we download automatically inside EviAnn if this option is enabled. And we provide labels on protein coding genes saying, okay, this is similar to such and such protein, or we provide a label that it's not similar to anything that's seen before. So in summary, EviAnn is all-in-one package, very easy to install. You can get it from GitHub, or you can get it now from Bioconda as EviAnn. It provides ultra-fast automatic annotation. It's validated. It annotates long non-coding RNAs, which not many other packages do. In fact, only Finder is the competing package that annotates long non-coding RNAs. It gives you UTR annotations for most protein coding genes. It outputs format compatible. I specifically designed it to output format compatible with stable to ISN tool for GenBank submissions. And my GitHub site has information on how to use that tool to submit, to create a submission package for EviAnn. It supports, the latest version supports mitochondrial genetic code. So you just give it a set of contigs to be treated as mitochondria in annotation, and it will annotate them slightly differently because in mitochondria, the genetic code is slightly different. In fact, start and stop codons are slightly different, so it'll take that into account. And it also supports all latest data. Illumina RNA-seq, Oxford Nanopore, PacBio IsoSeq, Kinnex, and mixed data sets. That is, for example, if you sequence the same tissue with Illumina and Oxford Nanopore, you can provide that as a mixed data set, and it will use data from both types of data at the same time. The latest release is available at GitHub, and I keep Bioconda also updated. So if you install it through Conda, you always get the latest release. And then finally, I would like to acknowledge my collaborators, especially Katerina Hoff, Alexander Lamzadze, and Mark Borodovsky. They're the leading authors of Breaker 3 software, and I learned a lot in conversations with them, and I got a lot of advice from them to develop EviAnn. And finally, I would like to acknowledge our funders, NSF and NIH, grants to Zimin and Salzburg. And now, thank you for your attention, and I take any questions that you may have. What if no closely related species has data available? typically the protein homology. So when I use protein homology, I'm not really looking for high quality protein matches. I'm looking to get splice sites out of protein alignments. And those are extremely well conserved, even if you use proteins from very distant genomes. For example, you can use proteins from Arabidopsis to annotate bristlecone pine conifer genome. They are very far apart on evolutionary on taxonomy tree, but you will get a lot of correct splice junctions. And in fact, I am using Arabidopsis proteins because there's a very nicely curated protein set to annotate conifer genomes. So yes, you can use proteins that are fairly distant and still get very good results. But of course, it does help if you have proteins that are closely related. So there's another question. How do you expect EviAnn to perform in species where only one or two RNA-seq data sets are available? Do you expect difference between long and short RNA-seq? Well, RNA-seq data, the more the better. If you have very little RNA-seq data, annotation, and annotation will not necessarily be complete. It also depends on the genome that you are working with. For example, for an insect genome, that's a few hundred megabases. One or two RNA-seq data sets will be sufficient. If you're working on a complex genome, such as a mammalian genome, 3 gigabase genome, one or two data sets will not be sufficient. Also for larger insects like spiders or whose genomes can reach into gigabases, you need to get more RNA-seq data. EviAnn is evidence-based, so the more evidence you get is the better. Now, in terms of long and short RNA-seq, up until recently, I've been recommending getting Illumina RNA-seq data because it was the most cost-effective and it had the least amount of problems in producing transcripts. But my recent experiments show that PacBioKinEx data can outperform Illumina RNA-seq. With the cost per gigabase about the same, it's about $10 per gigabase, I fully recommend you to use PacBioKinEx data if you can get your hands on it, on the tool to replace Illumina RNA-seq data. And so assemblies will be more complete and transcripts will be more correct. Also, question from Tom Brown. For many of the species we're working on in ERGA, even 6 data sets is a lot. How can we generate good annotations for our genomes? Well, you actually don't have to have RNA-seq data from your specific organism. You can use RNA-seq data from closely related organisms. You can download RNA-seq data from a close relative. For example, if you sequence a cow, you can definitely use RNA-seq data from different breed or even a close relative of a cow. Also, EviAnn allows you to not only use RNA-seq data, you can also use transcripts from closely related species, such as, for example, white bark pine and bristlecone pine. They're not very closely related. If you align the DNA sequence, it does align, but the alignment percentage is about 90, 94, 95%. And the alignments are not complete. But by adding transcripts from white bark pine annotation to bristlecone pine RNA-seq data, and EviAnn accepts transcripts from closely related species as a FASTA file with a special switch as an extra parameter, extra input. I got a better annotation. I got more protein coding genes. My BUSCO scores have improved. So in addition to RNA-seq data, if you don't have RNA-seq data, you can use transcripts from closely related species. Did you release your Drosophila melanogaster EviAnn annotation? No, I did not, but I'll be happy to send it to you. So I run it. This is something that I rerun every time I make an update. It runs automatically. I'll be happy to share. I'll be happy to share it, share the annotation or post it on GitHub. In fact, I can do that after the seminar. After the seminar is over, if you're interested, I'll be happy to share it. So another question from Jack Tierney. Is EviAnn limited to use an organism that uses the standard genetic code? For now, EviAnn only supports standard eukaryotic genetic code and mitochondrial chloroplast genetic code. Because typically, you assemble a genome, you assemble mitochondria. If it's a plant, you assemble chloroplast, and you want to be able to do this annotation in one run. So yes, EviAnn does partially support non-standard genetic code, only for now, only mitochondria and chloroplast. And to enable that, you just need to tell it, okay, these contigs in your input are mitochondria chloroplast, and it will annotate them accordingly. So another question. Secondly, how does in cases of recoding? Selena cysteine insertions and program or ribosome frame shifting. These are exceptions, and unfortunately, they are not going to be annotated. So EviAnn is a general purpose tool, and I can't account for all possible exceptions. If I start accounting for exceptions like selenecysteine insertions, I may, other cases that are errors may look like those cases. And I have to be very careful to avoid degrading performance on standard data sets to account for those non-standard cases. What quantity of proteins from related species do you recommend? How many organisms? So it's a question from Nora Jesslain. I recommend to get 500,000 to 1,000,000 proteins from related species. They're easy to get. And what I do and what I recommend you do is to follow instructions on EviAnn GitHub page with screenshots. And I can do a little demo here later on, where you basically go to NCBI taxonomy and you search for your organism. And then once you find it, and almost all organisms are in the taxonomy tree, you can keep going up the taxonomy tree and check how many proteins are available for this taxonomy level by looking at the number of proteins. And once you get to the number that's above 500,000 up to 1,000,000, you can just download these proteins from NCBI and just use them. So that's my recommendation, and that's what I do when I run annotation projects, and that gives me very, very good results. So 500 to 1,000 to 1,000,000 proteins, and just get them from NCBI protein database, NCBI, by following taxonomy tree up. If you are working so Marius Marstrad, if you're working comparative on, for example, an insect genus, is it a good idea to run iteration for all species, then rerunning for all species with resulted proteins for all species? I'm not sure how to answer this question. My first intuition is no, because EviAnn does make mistakes. You do not want to use errors to propagate. So I would rather, so EviAnn will find novel proteins. The venue to find novel proteins is when, for example, you have a chunk of a protein from related species that matches a transcript, and EviAnn is able to extend it for the entire transcript, potentially producing mostly new protein. Or sometimes EviAnn has algorithms to actually extend the transcripts and do a little bit of ab initio like process. It's similar to what GNOMON does, NCBI GNOMON does, and that may result in novel amino acid sequences. Also, EviAnn does use transdecoders, so for novel, it can find novel proteins, although the number of those novel proteins is usually very small. So and the error rate is pretty high. Just to avoid errors, I would recommend to simply use proteins that are available from NCBI. And note that these proteins are not necessarily in any of the annotations of these organisms. These proteins are sometimes only present as proteins in GenBank. And as I showed you in my Drosophila experiment. So the amount of protein data in GenBank is actually very, very large at this point, and it gets better all the time. So it increases all the time, and EviAnn utility will get better with time as more proteins are added to GenBank. Frederic, hello. When gene is from the same family and tandem on the genome, his annotation is often poor. Is EviAnn giving better results? If genes are next to each other on a genome, EviAnn would annotate each copy. And in fact, I'm actually aware that problems may happen. Like, for example, you get a transcript where you get a first exon from one gene and two exons from the other gene. I specifically have algorithms to prevent and disentangle that. I call these read-through transcripts, where a transcript goes and merges two different nearby genes. Sometimes it's a real read-through phenomenon, but most cases it's just a mistake. It's an error in alignment or transcript assembly, and I have algorithms to fix that in EviAnn. Yeah, so maybe we can, we can move on. I guess it's time to move on to a short demo of EviAnn annotation. So let me share a different screen. Because I'm not, or let's see if I can pull my other screen onto here. I think this is a better option instead of going to a screen. So here, I'm going to show you how to use EviAnn to annotate yeast genome. It's going to go very fast. So for yeast genome, I'm going to use data in this folder. So three key inputs to EviAnn are genomic sequence. This is genomic sequence for bread yeast, Saccharomyces cerevisiae. I got it straight from RefSeq database. Then I'm going to use just a set of proteins that I downloaded from NCBI, and here I specifically excluded the Saccharomyces cerevisiae just to avoid getting proteins that are from that particular genome, to avoid getting the right answer in my input data. And this is just a FASTA file of protein sequences. So just a FASTA file of proteins. Note that when you download proteins from GenBank, sometimes you get a label that tells you what this protein does and what species it belongs to. Actually, EviAnn, if this protein is used as evidence, EviAnn will preserve this label, giving you function kind of for free. You don't even need to do functional annotation. If your if your evidence has labels, it will preserve this label in the final output. And then finally, there is a file that contains all RNA-seq data. And the way I decided to implement this is it's a file that contains paths to your FASTA, FASTQ, or BAM files, and it's tab-separated file. If you, for example, if you just have Illumina RNA-seq data, you first give the location of the forward reads, tab, location of reversed reads. No need to put anything else. For other types of data, such as BAM files or mix files, you have to consult EviAnn usage message. Just type EviAnn-H, and it will actually tell you the format of this file. The format is very simple. It's, oops, excuse me, it's the file name with the path, and then there is an optional tag. And so there are several types of files. If you have a BAM file, for example, you just give a file name, and then the tag would be BAM. Here is the list of all tags that are recognized. So FASTQ tag is default. If you don't give any tag, it's assumed that it's FASTQ, but you can put in FASTA, BAM, BAM isoseq. BAM isoseq means that it's a long read, BAM file of aligned long reads. Isoseq, this means the data is PacBio isoseq reads in FASTA or FASTQ format. Or it could be Kinex. I don't recommend using Oxford Nanopore reads except for in mix mode. This mix means that the data from same sample sequenced with both Illumina RNA-seq data and long read data, such as Oxford Nanopore, and in this case, the EviAnn would expect 3 files. That is forward Illumina reads, reverse Illumina reads, and Oxford Nanopore reads, and then tag would be mix. And finally, BAM mix. It expects 2 files. First BAM file is Illumina BAM file. Second BAM file is long read BAM file. So this may seem like a lot, and yes, I'm going through this very fast, but this is all recorded in usage message, and I hope these instructions are fairly clear. It's available both on GitHub and just by typing EviAnn-H. So, but over here, we just have paths to 2 very small RNA-seq data, to 1 very small RNA-seq data set that I got from NCBI. And here I'm going to run EviAnn. So this is the command line, EviAnn.sh. You just have to run with the path to EviAnn. -R gives you the which is used for RNA-seq data file. So this is the file that has paths to RNA-seq data files. -G, you supply the genomic sequence, and with P, you supply the evidence from protein. You can supply evidence from proteins. And -T tells to use 16 threads. So currently there should be nothing in this folder. I'm gonna remove if anything was there, and then run EviAnn. It checks for dependencies. It makes sure that all dependencies are present. And most of these dependencies are distributed with EviAnn, such as UFASTA, string-tie, it's a static executable, GFF read, GFF compare. These are very useful tools to work with GFF files developed here at Hopkins. Miniprot, it compiles on install, and if you use Bioconda version, it'll just pull up the package. Transdecoder, also distributed with EviAnn. A modified version where I cleaned up some dependencies. And then the rest of the tools, such as Minimap2, HiSat2, some tools, Minimap2, HiSat2, they are required to be installed on the system. And then some tools and NCBI BLAST are also distributed with EviAnn. So the only 2 dependencies that you need to have if you are installing from GitHub is Minimap2 and HiSat2. And if you're installing from Bioconda, then everything will be installed. And so it checks for dependencies. It computes maximum intron size. Now, maximum intron size is a very, very important parameter. If you don't get it right, and it's important because it's a parameter to Miniprot, and it's also a parameter to HiSat. If you don't get it right, if you set it too small, then any transcript that's larger than that size will not be assembled. If you set it too large, then repeat regions may cause spurious ultra-long transcripts in your output. And EviAnn, we actually preparing a paper where we show the mechanisms for intron size, propose a mechanism for intron size evolution. And there is a model that EviAnn uses to compute the maximum intron size automatically based on the input genome size. And so, of course, you can override that in the parameters, but EviAnn will compute that will estimate that automatically for you. So here it's built in HiSat2 index, aligned RNA-seq reads. If you give EviAnn BAM files, it will skip those steps and it will just use BAM files to assemble transcripts. You don't need to merge transcripts from various different libraries or RNA-seq runs. EviAnn does this automatically. And so it aligns proteins, computes Markov chain matrices, and in just a short time, annotation will be done. So let me make sure that everyone can see the results. I'm going to move window to the center of the screen. So it's going to be probably another minute to output this annotation. It's a very small genome. It's only 12 megabases. And so over here, you see found misoriented transcripts. These are transcripts that are single exon transcripts. And if transcript is intronless, it's actually hard to figure out which DNA strand it comes from unless you have protein evidence. In the protein, you do have orientation. You do have strand information in the protein. Read-through transcripts, there are 0 in these data sets, and these are transcripts that spuriously may spuriously merge to proteins. So final step is detecting process pseudogenes. There probably will be none in this very simple organism. Actually, most of the genes are single exon or intronless genes. And so finally, EviAnn gives you some statistics. It found 5,920 genes. Most of them are protein coding. There are a few pseudogenes. Number of transcripts is larger than number of genes, so there is some alternative splicing here. And there are 5968 distinct proteins. So, and then now you can, for example, run GFF compare to evaluate how good this annotation is compared to GFF file that I downloaded from NCBI. This is a reference annotation for this genome. And then you can look at the statistics. And you can see that with this very small RNA-seq data sets, it's only about a gig of data. We were able to get 91%, almost 91% of transcripts with this precision of 97%. And we missed 624 exons. We introduced a few novel exons. But overall, this is the annotation, and we get about 92% of genes with a precision of 99%. So this is annotation of yeast genome with a very small RNA-seq data sets and proteins that excluded the proteins from this particular yeast. So that's, oh yeah, and finally, what does the output look like? So the output files are described on GitHub site. So this is what output looks like. So for EviAnn, you have gene, mRNA, exon, CDS. All these objects are here. And one thing that's particular to EviAnn is for every mRNA that it's protein coding, you have tags, evidence protein ID, and evidence transcript ID. The most important one is evidence protein ID, and this tells you this particular protein has been annotated by homology to this particular protein from Saccharomyces kudryavzei, related species. And it also tells you which transcript was used to annotate this protein. It tells you what start and stop codons are. By default, EviAnn only annotates complete proteins. That is, they have start and stop. You can relax that, and then EviAnn will annotate proteins that have either start or a stop, and that's an option. I don't recommend you using it. And it tells you the type of evidence that was used. So this evidence is protein only. That means that this particular gene would not have would just this particular gene or CDS and exon was annotated from just protein alignment. No transcript evidence was used here, and as you can see that there are no UTRs. Exon coordinates are equal to CDS coordinates, and then in fact, since I used very small RNA-seq data set to shorten the runtime, most of the proteins will be annotated using protein-only evidence. So another type of evidence here is evidence complete. This is where a transcript was used, assembled from RNA-seq data, was used along with protein evidence. And as you can see, there are UTR regions annotated. You can see that by seeing that exon starts earlier than CDS and exon ends later than the CDS. So the CDS here is a subset of the exon, and this is the complete annotation with UTRs. So I'm trying to make it user-friendly, and if you have any suggestions how to make it better, I can always do it. So the question is why no specific data type for ONT? As I mentioned, I do not recommend that you use ONT data by itself because of the high error rate. My only recommendation to use ONT data is if you have a mixed data set with Illumina and ONT, then you can use it as mix or BAM mix. And question from an guest line, can input files be in compressed format? Yes, they can be. I do zcat on all files. BAM files are compressed. They're all FASTA, FASTQ, could be either gzipped or not gzipped. It'll be fine. So EviAnn will automatically deal with compressed files.
Okay, thank you, Aleksey. I'm afraid we are at time, but I want to thank you for a fantastic talk and a very interesting demo, and also amplify really the user friendliness of this tool and how well it is working for us in the lab already. So thanks again for taking the time to show the details of your tool.
Okay, well, thank you for having me, and it's been my pleasure to present in the seminar. Thank you, and good luck. Bye.